Welcome everyone to this week's edition of the Extracellular Vesicle Club. We're very happy to have you here. And I'm also pleased to introduce our presenter today, who is Claudia Torrecilhas, who is coming to us from Brazil. And she's going to be presenting some recent work from her group that will include some interesting biomarker discovery and also some indications of function of EVs. And there's going to be some methodologic components to this as well. So I think this is going to be a very exciting journal club. Some of you may know that the Journal of Extracellular Vesicles just received a new impact factor today, and we were kind of surprised to see that it's gone up to over 25, in fact, 25.8. So I just wanted to say that, you know, the impact factor is not something that ISEV is working towards actively. It's not something that we want to necessarily misuse, but it is an indication, I think, of the tremendous work that many of us in the community have done. Many of you have done by contributing work to the journal. And I want to especially congratulate our current editor-in-chief, Jan Letval, and the previous editors-in-chief, Peter Quisenberry, Yang-Sung Go, and Clotilde Terry, who really helped to build that journal and get it to the state where it is today. So congrats, Jan, and all on that. And please, everybody, continue to submit your best work to the journal. So today is, I think, our first edition of what we're going to call a Meet the ISEV Leaders EV Club. And so Claudia, thank you for being here for this inaugural edition. So in addition to hearing about Claudia's work, I would just like to ask a few questions in the beginning so that people get to know a little bit about you and your background. So Anna, can you tell us, a little bit about your scientific background and how you became interested in extracellular vesicles?
Yes, I'm biological science. I finished university more than 30 years ago, and I started to work to transplant. Renal transplant is immunogenetics here in Brazil, because Brazil is the second place around the world to kidney transplantation. And my background started in immunogenetics, I like so much, but I need for new challenges in my scientific life. And I moved to São Paulo. Is like a country. Live here in São Paulo with 22 million people in the cities. I'm crazy cities. They never sleep. The city 24 hours. You find a lot of to do here, but now it's closed, of course, because the COVID pandemic. And I started to work to patients. I like so much in the renal transplant, but my ex-supervisor, my PhD, come to Dundee. He's a Brazilian. He lives in US now, Igor Correia de Almeida. He opened his lab. And I'm very excited because I sent a letter for him to propose, I'd like to PhD with you, to know your lab. And I start to work with him in Chagas diseases. But my project is my 2 years, I don't have any good result in my project. And I'm so upset, and I talked to Igor, and now my first supervisor and my first postdoc, Maria Julia, he started to work 2 EVs on protozoa parasite, and she traveled to European Society for Parasitology and showed these results about EVs, and the people didn't believe in her results, and she stopped to work 2 EVs 30 years ago. And Igor and Maria Julia started a collaboration for characterization for the molecules, the glycoconjugates in the parasites. And Igor, Anna, why not talk to Julia about it? Because our project is very difficult, or not to move to EVs. EVs, Igor, but it's difficult to know, but I don't know EVs. It's a particle, Anna. And here start to finish my PhD. Igor moved to US. He opened his lab in US more than 15 years. And Maria Julia and I started to work in my first postdoc. And when I finished my first postdoc, I moved to US to work in my second postdoc to half trip to virus. Influenza virus is very good background I have with half trip in the UGA. When I come back to Brazil, application for university. Now I'm associate professor, but I fall in love with this because it's very exciting fields to work. It's difficult, it's not easy, but this time to start my PhD, and now is more than 20 years to work in the EVs. One day, Maria Julia, my ex-supervisor, called me, Ana, why not to write this a review about EVs? This is a more than 20 years. And I start to review with her, and I read a second review with Maria Julia, and I founded the International Society for Extracellular Vesicles, and my first congress in Boston. And I'm very glad because it support our work. And I start here. I have a project, I have a lot of collaborations, and this is very exciting because I like so much to work to patients, and I like so much with EVs. And now this work with Rafael, my students for PhD, is very good because it's two fields, two areas, and I started to combine. It's very good. And
Yeah,
this
and
is my
it's so
life, scientific
exciting.
life.
It's certainly
Yes.
exciting to see how the field has developed in the time that you've been in it and all of the changes that you've seen over the years. So we're looking forward to hearing about your work in just a minute. But I wanted to also invite you to maybe say a few words about the society that you have founded for Latin America.
When my first congress in Boston is difficult congress because is a lot of problems in Boston because it's a bomb, and you stay for 4 days together in this hotel in Boston, in the downtown in Boston. And a lot of Brazilians started to work with me, and Hernando Del Portillo but Hernando from Colombia, but he lives in Spain. Why not, Ana? Because we started maybe our society in Spain. Why not in Brazilian? Because a lot of people work with EVs in Brazil or Latin America. And I talked to people here, and the people suggest us and work to move, not this Brazilian society more, but it's a Latin society. Because if you remember, Ken, in the Congress workshop in Buenos Aires, a lot of people work in Latin America, and it's not as big society, but we would like to, when I have a congress, is a big congress like this Genetic Society, is a Cell Biology Society, one day before is Education Day, and improve this Latin America society to improve and they learn more people how to work with this. This year is a cell biology congresses. The people invite me to organize this, but it's a virtual meeting. It's possible. But maybe next year when open the face-to-face, the congress started to these education days for each congress like this, and maybe one day organize. We invite you to come back to Brazil to organize this, the first meeting in the Latin America Society.
Well, that's very exciting to hear about what's being done in the Latin Society, and I know that the International Society for EVs is very supportive of this, and we look forward to strengthening the different chapters of ISEV, in this case the Americas, and seeing what can be done locally as well as on the international level. So, Ana, thank you so much for your leadership with ISEV and now with this society. So. I'd like to invite you now to share your screen and go ahead with your presentation. I'd like to ask everybody else to please put your comments and your questions in the chat box, and then at the end of Ana's presentation, we will unmute and allow some discussion. So go ahead, Ana.
Yes, and thanks so much for invitation me. Thanks so much for invitation me, Ken. This paper we publish for this year is Rafael Pedro Madeira, my PhD student. He finished next month his PhD, and Rafael and I started to work to EVs in Chagas disease. Why start to work EVs in Chagas disease? What the big problem to Chagas disease? And what the next step to understand the parasite-host interactions? And you have the collaborations for University of São Paulo is the big hospital and the biggest hospital in America, Latin America, in São Paulo, and the big, a lot of patients with Chagas disease. He's Dr. Charles Madi and Dr. Barbara Mariani helping me for select the patients to analyze the EVs. But is this new open, but is a promise to the new biomarker in Chagas disease, okay? And the Chagas disease is just for the introduction. What's the big problem now is the neglect tropical diseases. And a lot of protozoa parasites to infect the people in Latin America. But now the Chagas disease or American trypanosomiasis is spread around the world. And the April is day of the Chagas diseases, because this is a vector to transmit Chagas diseases, and I explain how the transmission this disease is the timeline with the first human case of Chagas disease report in Latin America. This is a girl. Her name is Berenice. In my lab, I have this sample of her parasite infectant. In the Brazilian, Carlos Chagas is a doctor, isolate the parasite, characterization the forms of parasite from Berenice, and after that, the phase of disease, acute and chronic phase, and a lot of to do. And the first evidence in the mummies in Chile isolate the Trypanosoma cruzi, the agent of Chagas diseases. And a lot of questions, a lot of discovery, and the big question is, vaccination these diseases or not? What happened? And is the big problem for Chagas disease. And the Chagas disease is spread around the world. Spread the US and Europe as well, and Japan and Australia. But how the disease is spread? The blood bank is not controlled. This blood contamination, the people to immigration for endemic areas move to Germany, move to Italy, move to Spain and US and Canada. But the triatom infestans, the vector, move the Mexico for US and increase 54%, 54% the infections in the US now. The Chagas disease now is a tropical neglect disease, but now is an emergent disease in the West because migration for chronic infections, people to endemic countries, and the blood banks is not screened for Chagas disease, and few exceptions, and the people spread the infection, and the people don't see it is parasite, the acute phase is asymptomatic just for a fever and the control, and after years, the chronic infections. And the big problem maybe is a transplant of heart is very, very big problem. This transmission is from arthrop, is a triatom infestans, Hodinium prolixus, is a 8%, but in Brazil is completely controlled the transmission for vector. And now in the north in Brazil, it's an oral infection, is a 10% for acai juice, sugar cane, and the orange juice transmitted this infection, okay? But the people don't see the vector and contain infective forms, and the people contaminate food or juice contaminate. And the mother to children is a 1%, and the blood 26%. And the organs is not have problem here in Brazil, but if you don't screen the blood or the organs, it transmits for another people. The infection is a big problem now and around the world. And there are only 2 treatments for chemotherapy for Chagas disease, nifurtimox and benzimidazole. Is not effective because a lot of side effects for people, and the treatment for 2 months is done under doctor supervisions, and the people took the pill and the parasite start to proliferation. Is it a big problem now? But you have the treatment, a lot of side effects. You need to more approach or vaccination or therapy or modulation, how the host immune systems start to new treatment or vaccination or therapy. Is a lot of questions for this. And you have more this. I don't explain is deep for life cycle or Trypanosoma cruzi, but pay attention this. These metacyclic forms in the vector, in the physis of vector, contain these forms, infective forms, and this is transmit this in the physis for human vertebrates people. And after that, as an intracellular parasite, this parasite stay in the blood in the acute phase and infect cells, and they spread for proliferative forms of mastigotes and transform in triple mastigotes forms. And after acute phase, move to heart, intestines, and another organs. And the big problem is for the heart effect. And the vector, when they feed for blood, start this new cycle and continues the infection. But now is a big problem is the blood banks, oral infections, and congenital infection is a big problem around the world. But the clinical course, this is a week. 1% of people is a heart or brain problems is usually in children's, but 100% is asymptomatic with normal organ functions and stay for years, and you have the indeterminate forms of Chagas diseases, 7% asymptomatic with normal organs function, but 20% of people is a dysfunctions for heart or intestines, and there's 10% symptomatic organ dysfunctions. But this is balance for immune systems is big challenge to understand the clinical course of Trypanosoma cruzi infection in human. On the most finding of patients is this cardiopathia is a cardiac modulation disease, is a myocard difficult because this parasite is still in the heart tissue, and the proliferation in the mastigotes nests, and the tissues, heart tissues, lose the functions, and the aneurysms, thromboembolisms, is the myocarditis is completely lose the functions, and the chronic phase, the people move maybe in the heart transplant or die. We have the hospital to improve the list of patients to Chagas diseases to heart transplant here in São Paulo. But a lot of problems after years, the infections of Chagas disease is not in acute phase. But in our study and in this USP or Unifesp Department of Cardiology, follow the New York Heart Associations to the score to the symptoms classification systems. But you have 4 types of patients, class or score is score 1, 2, 3, and 4. And the last months or the last years of Chagas disease is the worst problems, is the people start to lose job, or is couldn't to walk, and is very, very bad situation in when they finish his life with this disease in chronic phase. But my group starts to work in vivo and in vitro assays. We show the triple mastigotes forms to stay in the blood for patients to acute phase, and started to isolate these EVs. What these EVs contain? The parasite releases spontaneously EVs. These EVs contain the glycoconjugate like mucins, TCH5, and transalidase. But what these proteins, what the glycoconjugates modulate the parasite-host interactions? They spread infections, these increase the invasion of parasite in the host cells, modulate inflammation, increase the migration for cells for the microenvironment in the infections, and understand interaction toll-like receptor 2 and modulate this innate immune response and spread more infection. But parasite die, yes, but parasite is an intracellular parasite. When the parasite starts to infection, the parasite release EVs and spread more infections. It's good for parasite to release these vesicles, but what happened in infection in vivo? And we examined the effect of the parasite vesicles during the animal infection to understand what happened to heart in animals. For this is a cartoon, is a scan electronic microscopy. This is a human cells. This is a triple mastigotes form in red. This is in yellow is EVs. The parasite release these EVs. And how the experimental protocol, we inject the animal EVs, isolate from parasite after infections, and I looked parasitemia because you have, in my lab, you have the acute and the chronic model to understand, to study the Chagas disease in animal. But this experiment is very important because we analyze the histopathology analysis to see what happened in the heart. And as you can see here, the heart for animals pre-treated with EVs, we found a lot inflammatory infiltrate. This inflammatory infiltrate is macrophages, lymphocyte T, and as you can see here, it spread more infections. Here is a mastigotes nests, is population more parasite in the heart tissues. Here is a normal, is a control just for infections, not treated with Chagas disease. For us, it's a big challenge to understand why you have a lot of cells, why not to die the parasite and control the diseases. It's a big question to understand. And I don't know yet explain this, but we start to move to patients because the triple mastigotes forms release vesicles. These vesicles for proteomic analysis contain the almost glycoconjugates to interact toll-like receptor, induce the MAP kinase in the host cells, macrophages or monocytes, and induce the pro-inflammatory response, and these increase invasions of parasite. And my work and my group and the group of Dr. Rodrigo Soares work with the vector, and the vector modulate the EVs from epimastigotes forms, modulate this the interaction in the intestine of the vector. And the EVs is very important to invertebrate and vertebrate hosts, and this move to understand what happened for EVs, what happened the host pathogens interactions, is possible to use to therapy or vaccinations, and I move to patients. For this, my group, I have a lot of project to basic science, but Rafael Pedro Madeira, he started to clinical application, and I suggest him to use the EVs from past patients to see what happened, and it's possible to, is a biomarker to follow these patients in chronic phase, because it's impossible to find acute phase in the hospitals, because the patients not go to hospital, you have a fever, and just when they start to heart problems to move to hospitals. And Rafa and me started to write this grant, and Rafa started his PhD in my lab in the Federal University of São Paulo. And you have a lot of questions. This EVs-mediated the host interactions, and this act to control the acute or chronic infection, modulate the host cells interactions, is a virulence factor or not, is NDVs from patients or parasite is a potential candidate for therapy for neglect disease. Is possible to use this, and a lot of questions. And vesicles could carry the messenger or virulence factor to prime the cells and the parasite invasions, increase in these blood samples for patients, modulate or not the host parasite interactions. A lot of questions. For this is a Dr. Charles Madi from INCOR Hospital. He's a just for heart hospital, is the biggest in Latin America. This is a hospital. He is people with Chagas disease around the Brazil come to treat here, maybe move to São Paulo to treat it, because it's a free treatment for Chagas diseases. Is the transplant is free here in Brazil. And a lot of to do to understand the disease, because it's a long history about this neglect tropical disease. Now is an emergent disease in the US, Canada, Europe as well. And Dr. Charles Madi is select the patients for us and started to isolate the EVs to see what happened. And this is an age for female or male for patients, and I use the control without Chagas and the check for ELISA. And the age for patients to use, this is the oldest, this, and the clinical stage for indeterminate alterations for electrocardiograms and alterations and ventricular dysfunctions follow the New York Heart Associations. And you have for stage 1, 2, and 4, you didn't find yet any 3. Maybe the patients move the stage 1, 2, 4, and the big problem, because maybe this stage the patients or move to heart transplant or die. And this is a frequency of patients, is a balance to electrocardiogram alterations for and dysfunctions of ventricular dysfunctions. And this is a group to collect the samples for this work. And for this is collect the blood used for anticoagulant, DTA, citrate, or serum. And after that, use and you compare what the best anticoagulant or use the serums. For it, anticoagulant or serums is very good approach to isolate. Don't see any difference for size and concentration of EV isolates from patients and control. And they start to ultracentrifugations, and you have total EVs. After that, you use it to SEC, is a size exclusion chromatography to isolate EVs. And after that, because the EVs contain alpha-gal epitopes, is from mucins, and we have a specific antibody against EVs. And I use for ELISA to follow these fractions, and I use specific fractions we detect alpha-gal epitopes, is our markers to follow the EVs from parasite and from patients infection with Trypanosoma cruzi. And I use NYHA, and I use it to transmit electronic macroscopies to see the morphology. And this is a big challenge, because I follow a profile of SEC, and I use different anticoagulants and serum. And as you can see here in the fraction 8 and 16, you find positive from alpha-gal epitopes. And then I'm glad to see the result, because is the blood from patients is the mix of particles is very good, because you find these fractions and they start to work with these fractions. But just for improve the protocols, the methodology to use to propose these markers in the Chagas disease patients. And if you use anticoagulant, citrate, or EDTA or serum, is the same profile of SEC. Is very good, because is you stay very stable to reproducible. What kind of the vesicles, what kind of the population start to work. And here is a transmission electronic microscopy. As you can see here, a lot of integrity and quality of particles purification. And these cells from patients. And as you can see here, when the incubation for 24 hours or 48 hours, here's is the cells is very release vesicles too from patients, is in vitro assays. And it's very interesting, because the parasite is the same, maybe it's the nets or the nanotubes between one cell and two cells, and they start the communication. I saw a lot of this in the Trypanosoma cruzi in this scan electronic microscopy. And the next, we compare, Rafa compare if these EV profiles from patients with chronic diseases and controls, and the release EVs. As you can see here, is just for cells in attachment to the plates, incubation with the total parasite, and EVs isolate from parasite. As you can see here, the concentration of EVs from monocyte or macrophage, PBMCs, is lower than control, and the size is more 200 nanometers is if you compare with the controls, but the concentration decrease if you compare the control. But why the concentrations decrease? Why not? Because the parasite induces inflammation, but is this chronic phase, and I move to understand what happened with these patients. And I start to collect isolate EVs from plasma. As you can see here in NYHA, these concentrations is decreased than control, but when incubation, these EVs isolate from patients and with this human macrophage T8P1 in vitro assays, and collect this median and detect the cytokines, we found more interferon gamma and IL-17. Interferon gamma is very important to parasite to spread more infection, but this EVs isolate from past patients with chronic disease, Chagas disease, induce interferon gamma. It's very important. And these EVs contain alpha-gal epitopes and contain mucins. And mucins induce in vitro in our studies and the other groups to work in the Chagas disease, induce interferon gamma via mucins. And these EVs from patients contain alpha-gal epitopes and conclusion mucins is very important. And as you can see here is the frequency, because we stratifications the patients in determined forms, electrocardiograms, and electrocardiograms with these ventricular dysfunctions and control. As you can see, here's a difference for interferon gamma, and I couldn't see more difference in the IL-17. But here, as you can see, difference for frequency, the concentration values of interferon gamma. But the interferon gamma is the marker for these diseases. And in conclusion for our is a preliminary, we published but now we start to continue these studies to collect more periods, 1 month, 2 months, 6 months, 1 year, to understand if maintain the interferon gamma, if maintain the mucins in the EVs from the blood for patients. Maybe this timeline, maybe this proposed to start to work acute phase, but it's difficult to find patients with acute phase in the hospital, heart hospital here in São Paulo, and maybe is prepare the protocol to collect more samples for chronic phase and detect antibody cells and EVs to start to program to how to modulate the chronic phase and associate the EVs to isolate from patients with treatment. One that's reason to discussion, because you have acute and chronic phase, maybe the EVs induce interferon gamma is a base to inflammation, but the patients not die. But if you monitoring, if you follow these EVs during more years, maybe decrease the side effect of drugs, maybe propose the new therapy to follow the patients, and the quality of chronic patients of Chagas disease is better than other patients don't use the EVs to follow. But these EVs contain alpha-gal epitopes. It's a fantastic results, because it's a light in the tunnel, and you find maybe with EVs isolate from parasite, is decrease the dosage through the medicines, or maybe in the future to vaccination or therapy used to EVs, maybe you find the control and more stable patients with Chagas diseases. And I would like to thank you, the Federal University of São Paulo. Here's my place when I work. This Rafael, he's finished the next month his PhD, he moved to postdoctoral. He's a pharmacy student. My first student is my group. More the students finish, is a recycle my group now. This is, I would like to thank FAPES, PICAPs, and the CNPq for support. And I would like to thank your attention, and I would like to take your questions. Thanks so much.
Well, thank you very much, Anna, for that nice presentation. It was good to learn about Chagas disease and about some of the recent work that your group has done. So we do have some questions that have come in the chat box. I am going to allow unmuting now. So when I call on you, please be ready to unmute and to ask your question, and then feel free to also follow up as needed with Anna. So let's start with Susan, who has a question about the blood cell subtypes.
Yeah, and I think that you kind of answered this, but just to be clear, are there specific cells in the blood that are infected, and how did you determine that? Which cells were infected?
The parasite infected, the most infected for the epithelial cells, Susan, started the infection to epithelial cells. And after that, the parasite endothelium, and after that, the monocyte or macrophage. The parasite is an intracellular parasite, but the first cells the parasite infects is the epithelial cells. And after that, the acute phase, in the 1 month, is spread, and you have a lot of parasitemia, because in the monocyte, the parasite started to transform the mastocytes. One tripomastocyte infect the monocyte, is into the cytoplasm. You have the 500 mastocytes, and transform tripomastocytes and broke the cells and infect more cells in the blood.
So how did you determine that it was from the endothelial cells that it spread to the monocytes? What markers did you use, or how did you do that? Or how did you do that?
The cells, the marker cells, or the parasites? The marker, the cells.
the cells, how did you determine it was the monocytes that were primarily infected?
The primary infected the epithelium. Why? Because the vector, okay, when the vector transmits in the skin, and the first contact, the triple masculine metacyclic forms in the epithelial cells. After that, the spread for the blood in the circulation, the parasite, and after this epithelium is spread for the blood, because it starts after 1 month the parasitemia. The parasite starts the parasitemia, but the vectorial infection, the vector infection.
I understand the spread. I'm trying to understand how you determined, what was the experiment that told you it was monocytes that it was spreading to?
Because the cells to induce the immune response, the natural killers induce the interferon, is the first in the innate immune response. And after that, the monocytes macrophages is very important to efficient to more infection for the parasite. Okay. Because the parasite need more cells to infections, and the monocyte is orchestration this spread the infection.
It's okay. Thank you.
Welcome.
So this is coming from the basic biology then that's understood about the spread, and it's so I guess the markers, I guess, is what Susan was asking about. So
yeah, but the CD14 is the big marker. The parasite, the mucins. This not published this, but CD14 and toll-like receptor 2 and 4 is a good interaction for the mucins for spread this infection too.
So do the EVs have CD14 on them?
No, from parasite. Susan, for the EVs from parasite, you contain the mucins, TCH5, and transalidase. Is a glycoconjugate to interact and to invasion the host cells. Okay. But in the other hand, from the monocyte, you found the CD14 EVs.
Okay.
So do you think then that the host could host CD14 positive EVs be important in response then to the infection?
Yes,
because maybe. It could be that there's kind of a target there that would soak up some of the infection.
But you have two ways the parasite infections, monocyte or epithelial cells: invasions or interact with receptor. When the candidate is a receptor of toll-like receptors, maybe 2, 6 is because the dimer, and toll-like 4, and maybe participate the CD14. Is a hypothesis, but I need to more work to understand these mechanisms.
Great. Okay. Susan, did you have anything else to follow up with?
No, that's okay. Thanks.
Great. Okay. Well, thanks for the questions. Let's go now to Dolores DeVizio. Welcome, Dolores.
Hi, Ken. Hi, Anna. Nice to see your work. Thanks for sharing that. I have a general question, probably along the same lines of what was being discussed. I was wondering if you think that beside these mucins, there could be some biomolecule that is present in all EVs from parasites or eukaryotes that could induce this interferon gamma response, because we see the same thing as I mentioned to you some time ago when we use cancer-derived EVs in stromal cells. Mm-hmm. So what about like nucleic acid, like double-stranded RNA or DNA? Do you think that's something that could induce this response?
Yes. In the proteomic analysis, you found the mucins. These mucins is very important to induce the host immune response. We didn't find any DNA in these EVs isolate from parasite. For patients, I don't know yet. Okay. And we found RNA in these EVs isolate from triple mashed goat forms. And I contact you, Ken, and maybe when I start to travel, we sequence for RNA from EVs isolate from parasite. But we found RNA, but I need more studies, I need more experiment too. But this EVs contain 6% the glycoconjugates from parasite membrane and modulate this host immune response.
Okay, thank you.
Welcome.
So as I guess a bit of a follow-up to that, what in the body of the host, I mean, what percentage of parasite-derived EVs do you think are there compared with the total EVs? I mean, is this like a sizable percentage of the complement, or rather a very small needle in the haystack?
It's a good question, because the parasite after acute phase move to organs and stay there to proliferation in the mastigotes into the cardiac cells. But I don't know what the percent of the EVs from parasite. My students, André Cronenberg, we published a paper to use monocyte and macrophage because the cells to help to spread these infections. And when you infect the macrophage with Trypanosoma cruzi, and after 48 hours, and you collect the EVs isolate from these cells, and you move to proteomic analysis, you find is a little, but you find these hits from parasites. We found transialidase because this glycoconjugate is very important to escape the parasite for invasion and escape for vacuole to broke the parasite, the lysosomes. And we found the mucins too. But the percent, the EVs from parasites, I don't know. And maybe in the acute phase, I suppose you have more EVs from parasite because the parasite circulation, but the circulation is very quickly because it's a 1 or 2 months, the acute phase, and the after move to chronic. When there are reasons, you have the low EVs because you have the base inflammation, but it's not the high inflammation because the inflammation more specific is in the organs. But the percent, how many vesicles from parasite, I don't know yet. But the cells, in fact, release vesicles and contain glycoconjugates from Trypanosoma cruzi.
Yeah, it sounds definitely like a very difficult question to answer once you get into that chronic phase, because of that local, it's the local interactions in the tissue that are happening with those EVs, and that's very difficult to make.
One of the reasons is as a proposal to Dr. Charles Madi from INCOR USP São Paulo to collect 1 month, 6 months, 1 year, 3 years, 4 years, and compare during the chronic phase, because the patients lives more than 40 years, 50 years with the chronic phase.
Yeah,
but the heart is
very interesting study.
Yes,
yeah, that'll be
a big study.
Very good, very nice. And we have a few technical questions here too. So Haifa Al-Zahrani asks, and I believe that Haifa may have had to leave. Is Haifa here? I'm just checking the list. No. Okay, so what is the concentration of EVs that you use to treat your PBMCs in the in vitro experiments?
10 to 8.
How do you determine that, I guess? Yeah.
Yes, but 10 to 8 particles per ml.
10 to the 8th per ml of culture.
Why? Because I use the system to C8O cells transfectol-like receptor, and I prepare the different concentrations of particles to interact physically with toll-like receptor 2 or 4, and finally the cells and the use to flow cytometry to see the interaction toll-like receptor. And 10 to 8 particles per ml I use, and I use the monocyte and the macrophage, and works very well for us.
And a similar concentration then is from Neelam. Neelam, do you want to ask your question? This is about the in vivo work, right? Neelam might not have the microphone on. So this is about, what about the, the concentration of EVs in the mice?
In the mice, and if you, because you have a lot of different strands for Trypanosoma cruzi, I use virulent strands, Y strand. And if you inject, because some in vitro assays to infection, if you pre-treat EVs, I use, for example, 10 to 5 parasite. I use 100 times more vesicles, these assays. For example, if you infect 2 parasite with 10 to 5 forms of Trypanosoma cruzi, I use 100 more EVs to infect, to pre-treat the animals. Do you understand? The MOA to the difference. 10 to 100 is 10 to 5 the parasite, for example, and use 100 more EVs for in vivo assays.
Okay, so you would go, you just go up by 2 orders of magnitude.
10 to 7.
Yeah, okay. And you were determining, that was based on concentration as determined by nanoparticles.
Yes, I suggest to prepare the first experiment, the kinetic, the time to interactions, and prepare inject many concentrations to find this, to find the best methodology to use in vivo.
So do you have any evidence, or if not evidence, then maybe some speculation about how long these parasite-derived EVs might persist in the organism? Because there are different estimations from biodistribution studies of the half-life of EVs once you inject them into mice, of course, in different ways. And there is, I think, an ongoing question in the field about whether the cell type that these EVs come from influences that half-life. And here, of course, you're injecting something from a completely different and very distantly related organism. Do you think that half-life in vivo is going to be shorter or longer than that of a mammalian EV that you might inject?
Is EVs from Trypanosoma cruzi, if you incubation EVs marker to lipid markers fluorescence and inject an animal, or if you incubation for the parasites, in 30 minutes, the other parasites, the experiment to interaction, to uptake in the first 30 minutes, the EVs is still in the other parasite. And if you isolate EV from other cells infected, if you incubation with the macrophage, in 15 minutes, these EVs uptake in the cells. And in vivo, I try several times to inject the labelly EVs, and I don't know if it's the concentration of these, but I don't have good result yet. But we found after 48 hours in the spleen, in the blender, but it's not as good a result. I need to improve this protocol, increase the number of years, or change the label to find the follow what the organs, what happened. But in vitro, we found this, the parasite communication, one parasite communication, the other parasite, and the EVs in the 30 minutes, you found the marker in another parasite. Okay,
I see. Yeah, so that's interesting. You do see a difference then between the parasite and the host EVs, though, in the in vitro experiments, and certainly those in vivo distribution experiments are difficult to do. There was a recent systematic review that appeared in JEV, I think just last week, on in vivo biodistribution studies So if anybody's interested, please go check that out. That was from Larry Chamley's group, and some others contributed to that. So.
But we have the parasite to transfect to tomato and GFP.
Yeah. Yeah. So
maybe this approach is very good to infection the animal, or I need to prepare the pilot, the good pilot, to understand the EV's distribution in the animal model, but it's difficult. It's not easy.
Right. Right. And you need a lot of material, so for sure.
Yes.
Yeah. All right. Well, it looks like we're coming to the end of the hour then. And I don't see more questions in the chat, but if anybody does have questions for our speaker, please reach out to Anna directly.
Yes.
So Anna, thank you. I think it's really just been a privilege for all of us to hear from you about this exciting work that you're doing. We wish you the best as you continue with it, and we will be glad to learn more about this system as you gather more data. So thanks again for joining. Thanks everybody for your questions and for your attentiveness today. If you do have any suggestions about future EV clubs, please reach out to me. I know there are a lot of interesting papers coming out, and we'd love to feature some of these. So thank you all. I hope you have a good rest of the week.
Thanks so much.
I look forward to seeing you soon. Bye now.